in the last video we have studied one of the application, biotechnological application in agriculture, how to make insect resistant plant. So in this session we are going to study the another application, biotechnological application in agriculture, how to make pest resistant plant. So let's start. See, one of the pest, one of the nematode, it is called Melodigyne incognitia. It's called Melodigyne incognitia. This is one of the nematode. It is one nematode, Melodigyne incognitia. It lives in the root of the tobacco plant. Where it lives? It lives in the root of the tobacco plant. What happens? It makes something swelling and it affects the root. Because of that, the plant cannot absorb water and minerals. So if the plant is not absorbing water and minerals, what will happen? Immediately the plant will dry. It will die. So this happens in the tobacco plants. Once one farmer, one person, he made a field. He cropped a plant contained in the field full of tobacco plants. What happened? He was doing hard work and the plant is growing day by day. But at one day, immediately at the morning, he came and he looked at these tobacco plants. What happened? All these tobacco plants become died. It means it is dried, damaged. That he searched, he finds what's the reason. He got this is nothing. It's because of the nematode that is called Melodigyne incognitia. So where it lives in the root of the tobacco plant. So now we have to make the plant resistant against this nematode, against this Melodigyne incognitia. So one of the process to make the resistant against the nematode, this Melodigyne incognitia, the process is called RNAi or the process is called RNA interference. So as we know, we have studied about DNA. DNA replication, DNA making is its own copy, that we can say replication. After replication, that copy, genetic information, it is copied into RNA, that we can say transcription. After transcription, the protein will be made, that we can say translation. So if the mRNA is made, then the protein will be expressed. It can express the character. So what we have to do? We have to silence this mRNA. Before formation of mRNA means silencing the mRNA. So that process only you can say RNA interference. So RNA interference we can achieve by the help of the mechanism that we can say cellular defense. The mechanism is called cellular defense. And the cellular defense is achieved by the help of the process that you can say silencing the mRNA. The process we can say silencing the mRNA. So how to silence the mRNA? We have to make double-stranded RNA. Double-stranded RNA. So what happens here? First of all, we can study what is the source of this double-stranded RNA. As we know, RNA is single-stranded, but here we have to silence this mRNA, we have to make double-stranded RNA. So what is the source of double-stranded RNA? You can understand, we have to infect a bacterium containing RNA to the plant. So it becomes transposons, mobile elements, it becomes the double-stranded RNA. So that we can say the source of double-stranded RNA. Now what we are going to do? We are going to make pest resistant plant. How it is possible? We have to make a vector. We have to take a vector, that is by the help of agrobacterium. So the plasmid we have to take agrobacterium and particular gene which is responsible to kill this nematode, that we have to insert into this agrobacterium. So the plasmid or the vector is ready. Now this vector is inserted into the plant. Agrobacterium vector, it is inserted, agrobacterium plasmid, it is inserted into the plant. That plasmid, it will make in the plant like sense and antisense, RNA. Sense and antisense RNA. So it becomes complementary with each other. It becomes double-stranded RNA. complementary with each other, double stranded. It becomes the double stranded RNA. So this double stranded RNA, it will be there in the plant. When the nematode comes, it will act to this nematode. This double stranded RNA, it is used to kill this mRNA. So by killing this mRNA, nematode cannot attack. It cannot attack to this tobacco plant so that nematode will die. The tobacco can be saved. Tobacco plant can be saved. It can be alive. So this technique we can say pest resistant plant. So this also we can understand one of the application of biotechnology in agriculture, that how to make pest resistant plant. So these are the application of biotechnology in agriculture. In the next session we can study how this biotechnology it is applied in medicine. It's very interesting topic. We can study in the next video. Thank you, students.